Kami ucapkan selamat datang kepada Bapak Ibu pada acara Guest Lecture Pusat Riset Veteriner, Badan Riset dan Inovasi Nasional, baik secara luring di KST Soekarno Cibinong, secara virtual melalui media Zoom, maupun melalui kanal YouTube BRIN Indonesia. Baiklah, acara akan kami mulai. Good morning, selamat pagi, dan salam sejahtera untuk kita semua. Yang terhormat Kepala Organisasi Riset Kesehatan, Badan Riset dan Inovasi Nasional, Profesor Dr. Hewan NLP Indi Darmayanti MSI. Yang kami hormati Kepala Pusat Riset Veteriner BRIN, Dr. Hewan Harimurti Nur Aji PhD. We are honored to welcome Dr. Adnan Alrubeye from University of Arkansas. Yang kami hormati moderator pada hari ini, Dr. Hewan Edi Sukmawinata, MSc PhD, peneliti ahli muda Pusat Riset Veteriner BRIN, para periset di lingkungan BRIN, serta peserta webinar yang berbahagia. Alhamdulillah, puji syukur kita panjatkan karena berkat rahmat Allah Subhanahu wa Ta'ala, Tuhan Yang Maha Esa, kita dapat bertemu dalam acara guest lecture dengan tema Broiler Chicken, Lameness, and its Implication pada hari ini, Senin 23 Desember 2024. Baik Bapak Ibu, mengawali acara pertama, kita akan mendengarkan secara hikmat lagu Kebangsaan Indonesia Raya. Kami mohon kepada Bapak Ibu untuk mengambil sikap sempurna. Baik Bapak Ibu, supaya acara pada hari ini berjalan dengan lancar, marilah kita berdoa bersama yang akan dipimpin oleh Dr. Hewan Aswin Afif Khairullah MSI, Periset Pusat Riset Veteriner. Kepada Bapak Aswin, kami silakan.
according to the guidance of the Islamic religion. Bismillahirrahmanirrahim. Allahumma sholli ala sayyidina Muhammad wa ala ali sayyidina Muhammad. Walhamdulillahirabbil alamin hamda syakirin hamda na'ilmin hamda yu'afini amahu wa yukafi mazida. Ya Rabbana lakal hamdu kama yambagi lil jalali wal ijil karimi wal adimi sultanik. Allahumma ya Allah, tiada kata dan rasa yang pantas kami haturkan kepadamu selain syukur kami kepadamu atas segala nikmat dan hidayah yang engkau anugerahkan kepada kami, sehingga kami bisa hadir bersama untuk mengikuti quiz lecture Broiler Chicken, Lameness and its Implication oleh Dr. Adnan Ali Khalaf Al-Rubaye di Pusat Riset Veteriner pada pagi hari ini. Allahumma ya Allah, jadikanlah acara kami sebagai majelis ilmu yang bermanfaat, yang engkau ridhoi, yang membawa barokah, rahmat, taufik, dan hidayah-Mu bagi kita semua. Ya Allah yang Maha Pengampun, Engkau Maha mengetahui terhadap kelemahan-kelemahan kami, terhadap kekurangan-kekurangan kami. Maka dari itu kepadamu lah kami menyembah, kepadamu lah kami mohon ampunan dan pertolongan. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Subhana rabbika rabbil izzati amma yasifun wa salamun alal mursalin walhamdulillahi rabbil alamin. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Baik Bapak Ibu, berikutnya sambutan sekaligus pembukaan oleh Kepala Organisasi Riset Kesehatan BRIN kepada Profesor Dr. Hewan NLP Indi Darmayanti MSI. Kami silakan.
Halo, dengar suara saya?
Sudah terdengar Ibu?
Baik, maaf tadi terlempar saya. Selamat pagi, Assalamualaikum warahmatullahi wabarakatuh. Very good morning all of you. Yang saya hormati Kepala Pusat Riset Veteriner DRH Harimurti Nuraji PhD, to honorable our guest speaker this morning, Dr. Adnan Ali Khalaf Al-Rubaye, Assistant Professor of Poultry Microbiology, Associate Director of Cell and Molecular Biology, University of Arkansas. Bapak dan Ibu Kepala Pusat Riset di lingkungan Organisasi Riset Kesehatan BRIN, para peserta webinar baik di lingkungan Organisasi Riset Kesehatan BRIN atau Bapak Ibu peserta dari berbagai institusi lainnya, sivitas organisasi riset kesehatan, dan Bapak Ibu yang hadir baik secara daring dan secara luring pada pagi hari ini. Alhamdulillahirabbil alamin, segala puji bagi kita panjatkan kehadirat Allah Subhanahu wa taala, karena atas berkat dan rahmatnya kita dapat berkumpul bersama dalam keadaan sehat walafiat untuk mengikuti acara hari ini, yaitu guest lecture dari Dr. Adnan Ali Khalaf Al-Rubaye dari University of Arkansas yang berjudul Broiler Chickens, Lameness and its Implication. Bapak Ibu yang saya hormati, Indonesia merupakan salah satu negara produsen unggas terbesar di dunia dengan sektor peternakan ayam broiler sebagai salah satu penopang utama industri protein hewani. Industri perunggasan khususnya ayam broiler di Indonesia telah mengalami perkembangan yang pesat dalam beberapa dekade terakhir. Ayam broiler kini menjadi salah satu komoditas unggulan yang memainkan peran penting dalam memenuhi kebutuhan protein hewani masyarakat Indonesia. Namun isu kesehatan ayam broiler khususnya lameness atau kelumpuhan menjadi tantangan yang signifikan dalam meningkatkan efisiensi produksi perunggasan baik dari sisi kesejahteraan hewan, produktivitas, ekonomi maupun kualitas produk. Kelumpuhan pada ayam broiler memiliki dampak yang luas dan kompleks yang mempengaruhi banyak aspek termasuk biaya operasional tentunya hingga reputasi dan keberlanjutan industri itu sendiri. Dalam hal ini tentunya peneliti memainkan peran yang sangat penting dalam mengatasi masalah kelumpuhan pada ayam broiler dengan pengetahuan yang mendalam dan pendekatan ilmiah dapat membantu tentunya menemukan solusi untuk mencegah, mengidentifikasi penyebab dan mengurangi dampak kelumpuhan pada ayam broiler. Selain itu, penting juga bagi peternak untuk memperhatikan faktor-faktor dari kelumpuhan tersebut seperti bagaimana manajemen pakannya, kemudian kondisi kandang, serta kesehatan ayam secara keseluruhan. Upaya pencegahan dan pengelolaan yang tepat dapat membantu meminimalkan risiko kelumpuhan dan meningkatkan efisiensi serta keberlanjutan dalam industri perunggasan. Bapak Ibu yang saya hormati, pada kesempatan ini kita mendapat kesempatan untuk mendalami isu yang sangat krusial dalam industri perunggasan, yaitu kelumpuhan pada ayam broiler itu sendiri. Bantu peternak menemukan solusi yang lebih baik untuk mengurangi kelumpuhan, meningkatkan kualitas daging ayam, dan juga mengoptimalkan keuntungan dalam industri ini. Melalui seminar hari ini, kita berharap dapat menggali lebih dalam penyebab kelumpuhan pada ayam broiler, termasuk faktor genetik, lingkungan, serta manajemen pemberian pakan. Kita juga akan membahas dampak jangka panjangnya terhadap kualitas hidup ayam dan implikasi pada rantai pasokan industri perunggasan. Oleh karena itu, kunjungan Dr. Adnan Rubaye dari Universitas Arkansas ini akan memberikan kesempatan penting untuk memperdalam wawasan terkait topik ini, sekaligus menjalin kolaborasi antara institusi
Mohon maaf, Bapak Ibu, sepertinya Prof. Indi keluar dari Zoom. Kita tunggu dulu sebentar.
mohon maaf, saya terpental lagi ini. Baik, ingin menyampaikan penghargaan dan terima kasih yang sebesar-besarnya kepada Dr. Adnan atas kesediaan waktunya untuk berbagi ilmu dan pengalaman dalam pertemuan ini. Terima kasih juga saya sampaikan kepada seluruh peserta manfaat yang besar bagi kita semua, memperkaya wawasan, mendukung upaya kita bersama dalam
Sekali lagi mohon maaf Bapak Ibu, terlempar kembali. Baik, kita tunggu sesaat lagi.
Halo, tes.
Baik, Prof.
Ya, baik, maaf ini. Baik, dengan mengucap Bismillahirrahmanirrahim, acara guest lecture, Broiler Chickens, Lameness, and its implication ini saya buka secara resmi. Semoga kita semua mendapatkan wawasan yang bermanfaat dari seminar hari ini. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Baik, terima kasih kepada Profesor Indi Darmayan atas sambutannya dan telah membuka acara pada hari ini. Baik, Bapak Ibu, acara selanjutnya kita akan melakukan sesi foto bersama. Kami mohon kesediaan Bapak Ibu di ruang panelis untuk buka kameranya dan memberikan senyum terbaiknya. Dan panitia akan membantu untuk melakukan tangkap layar. Baik, sudah siap Bapak Ibu. Saya mulai. 1, 2, 3. Baik, terima kasih Bapak Ibu. Selanjutnya kita memasuki sesi pemaparan dan diskusi yang akan dipandu oleh moderator pada hari ini, yaitu Dr. Hewan Edi Sukmawinata, MSc PhD, peneliti ahli muda Pusat Riset Veteriner BRIN. Kepada Bapak Edi Sukmawinata, kami silakan.
Baik, terima kasih Bu Yeti. Good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Honorable Chairman of Research Organization for Health, Professor Dr. Hewan Indi Darmayati, MSI, then Honorable Head of Research Center for Veterinary Science, Dr. Harimurti Nuraji, and then dear Honorable, today is our speaker, Dr. Adnan Ali Khalaf Al-Rubai. And then all of our audience who will join in Zoom and also offline in this room. So let me guide today's guest lecture that will delivered by Dr. Adnan Ali Khalif Al-Rubai. He is Assistant Professor of Poultry Microbiology and Associate Director of Cell and Molecular Biology from University of Arkansas. In today's lecture, he will show us the topic about a broiler chicken lameness and its implication. And before we start his lecture, I will introduce you the CV from the Dr. Adnan. Can the committee show his CV profile? Yes, Dr. Adnan graduated and complete his bachelor until master degree in Baghdad, Iraq, and then he continue again the master course and then completed the PhD course in University of Arkansas for the medical microbiology and continue for cell and molecular biology. And after that, he continue his academic and research profession as Associate Director of Graduate Program in Cell and Molecular Biology, and then Assistant Professor in Department of Poultry Science, University of Arkansas. And when I read his CV, most of his research topic about bacterial genome and vaccine related
Ada mic-nya.
I was born in Iraq in 1977. I was 3 years old, and Iraq had a very lengthy war with Iran for 8 years. Some of you guys may remember a long war between Iraq and Iran. And then I left Iraq in 2008 with my wife and 3 children. I got before that my bachelor from the College of Veterinary Medicine. So I'm a veterinarian from the College of Veterinary Medicine, University of Baghdad. And I ranked #1 among my colleagues, students, when I graduated. So I got a good opportunity to go to the University of Arkansas. After finishing my master's at the University of Baghdad, I went to the University of Arkansas and completed a second master's and then my PhD. And I have been working on chicken lameness. As Dr. Harris said, this has been the longest trip of my life. I'm an international traveler, so I travel to so many countries, talking about my research topic and trying to recruit students to the University of Arkansas. And so this trip has been the longest, but the moment I touched here in Indonesia, I've been very impressed by the kindness of the Indonesian people and how tasty the food is. For 2 days, I've been enjoying all different kinds of great food. We'll be talking about my research program, but before that, I want to talk about the University of Arkansas quickly. So I'm Associate Director for the Cell and Molecular Biology program at the University of Arkansas, which is our largest doctoral program on campus. We have over 100 doctoral students in the program. The University of Arkansas has 33,600 students. We are the flagship campus, the largest university in the state of Arkansas. And anyone of you guys know where Arkansas is? Have you heard about Texas? We're near Texas, side by side. Oklahoma, side by side. Missouri, side by side. So we're surrounded by all these states. But again, we're the chicken capital of the US. We have plenty of chickens. We produce 1 billion birds annually in Arkansas. We're also, our state is the #1 producer of rice in the US. I know you guys like rice. So we're the #1 producer of rice. In fact, the Minister of Agriculture for Indonesia visited Arkansas a few months ago, back in September, to sign some memorandum of agreements. We are very famous because we are the headquarters for Tyson. Anybody knows Tyson? The largest producer of meat for human consumption. We're the headquarters in our, within 20 minutes, headquarters for Walmart, headquarters for JB Hunt, which is the largest truck company in the world. So with all these companies, they keep giving good amounts of money to the university. JB Hunt last month gave $120 million US dollars as a gift to the University of Arkansas. The Walton family, Sam Walton, the Walmart, gave the university $300 million US dollars several years ago. So they keep donating to the university, and this is how we improve the university. So I'll be talking with you guys about the chicken lameness, which is when birds start becoming lame, and that's mainly because their size gets very, very big on the small legs, and this is how they develop lameness. So you will see all these bones in my presentation. Even if you're gonna have chicken for lunch today, go ahead and have chickens. I hope those pictures will not prevent you from having chicken for lunch if you're having chickens for today. So anyway, this is, we call it BCO, bacterial chondrocrosis with osteomyelitis, BCO lameness. So it's a bacterial reason. You can see all these birds sitting, their legs are extended and able to stand. And usually around 3 to 5% of birds, they get lameness. The problem with lameness is you don't, sometimes it's, you see the lameness and birds are limping, so you can say they are lame, but the majority of birds don't show lameness. They have it inside, they are lame, they are infected, but you don't see the infections until you cut them open and you start seeing them. So usually it's around 3 to 5% of birds they get lameness, and sometimes 20 to 25% whenever there is a widespread infection. In Arkansas, we produce 1 billion birds. In the US, we produce 9 billion birds per year. Worldwide, there are 25 billion birds produced annually. Chickens, broiler chickens for meat, 25. So we produce really a large amount of birds, and this contributes $2.8 billion to the economy of the state of Arkansas, where I am from. You can see from back in the days, 1960s, there was really huge focus on bovine and pork. But now you can see chickens, the green line going all the way up, because you can produce chickens in 42 to 45 days. So it's a quick turnaround to produce all these different kinds of meat. Now, why does lameness happen? So let's take a look right here. So this is 1957 at the top. You can see the chick weight was 34 grams. In 2005, it was 44 grams. So chick's size became bigger. You go all the way down. After 56 days, the bird size was 905 grams. That's in 1957. 1978, 1,800 grams. 2005, it's 4 kilograms. That's in 56 days. So you can see the size is 4 times in 2005 compared to the back days. Why? Because there is lots of genetic improvement. So they pick the large birds. There is also diet improvement. We're giving birds calculated diets, specific amounts of minerals, proteins, and everything. And of course, we're improving birds management. So I want to step back here. You see how big the bird right here, the small bird, compared to this large bird. If you look at their legs, the size of legs didn't differ. Still small legs. So with this large size on a small legs, this is going to put more pressure on the legs, and this is how birds will become lame. Lameness is the #1 animal welfare issue worldwide. It's the #1 disease affecting the poultry industry in the United States. We don't have in Newcastle. Again, we do have lots of chicken lameness. We have avian influenza, of course, but we don't have, again, some of the diseases that are widely spread. So with the large growth of birds, you will see all these different kinds of diseases. So 4 of them, sudden death syndrome, so birds are dead and they are flipped on their back. Ascites, you can see, we call it water belly, lots of fluids in the belly. And then, of course, leg disorders. So birds are sitting and unable to stand. With leg disorders, we have all different kinds of cases. So you start from here, that's rickets. Tibial dyschondroplasia. So if you slice the tibia, this is what you will see. BCO lameness, which is what I'm studying, foot dermatitis, twisted legs. All these cases are actually impacted by the large size of the birds. Because bird size is really big, this is how you will see all these cases. How many of you guys have seen some of these cases before? Raise your hand, please. How many of you guys have seen some of these cases before in the field? I guess everyone, maybe? Hopefully, okay, sounds good. So, talking about BCO lameness, those are the symptoms. You can see the bird is sitting, tired, the feather is ruffled. Sometimes they sit on their back. In fact, they have a name for this one. It has multiple names. We call it kinky back. Sometimes they call it the Harley Davidson. You know how the Harley Davidson motorcycles, where the riders, they have their legs up? This is called the Harley Davidson ride. Or you can see the wings are extended. Wings are extended, legs are like this. So all of this is called, again, BCO lameness, but it has different forms. So it's either it's affecting the legs or also the spinal cord. Can somebody click on this one, please? There's a video right here. No, with the mouse, because this is a video. Hold on. Yep. So you can see the birds will be sitting, unable to stand. And when you move them, so this is me holding this broom, trying to move them. And they just want to stop, walk for a few steps, and then they will sit. And in the most advanced cases, this is what you will see. You can see the bird is completely sitting, unable to stand because of the infection in the bones. Now, those birds should not be consumed. You cannot eat them. Why? Because those birds are full with bacterial infections, and those birds must be condemned, must be decomposed, and then just removed from the flock. So, why BCO lameness and why it's very important? Well, it's an animal welfare issue. Imagine you have an infection right here in the femoral head or the tibial head, and those bones are infected. How much pain is this? Too painful to those birds. So this is an animal welfare issue plus an economic loss. The main problem with lameness is that it doesn't happen during the early stages of development. Usually happens when the birds are really big after 40 days. So you're spending all this money on feed, management, vaccines, and then you will have to remove those birds. It's also a food safety issue. Why food safety? Again, because those bacteria will be in the blood and they are spread through the entire body of those birds. Let's see how it happens. So this is a tibial head. So if you get a knife or a blade and you cut the tibial head, this is you will see. This area right here is the most important. We call the feces or the growth plate right here, which is the area that will be mostly affected by lameness. Let's look at the bone. So this is a day-old chick, 40 days, 40 grams. And this is day 42, which is around 3 to 4 kilograms. You can see there's a development in the bone, but it's not really matching the increase in the size of the bird, in the muscles of the bird. So with this increase in the size, it's going to put too much pressure on the growth plate, and this is how lameness will happen. So there is too much pressure on femoral heads, tibial heads, and again, those birds are unable to withstand all this pressure, which will eventually lead to the damage of the growth plate, leading to lameness. All right, so this is GP, which is the growth plate. Now, if you remember from a few minutes ago when I said we have kinky back cases, the Harley Davidson, this one is actually affecting the growth of the flexible thoracic vertebrae. Vertebrae number 4, the thoracic vertebrae number 4. This one is affected. Why? Because the air sacs in the bird are connected to the flexible thoracic vertebrae. And every time those birds breathe air inside, this air will actually go inside and gets to the flexible thoracic vertebrae, and this is how it will lead to the infection, leading to kinky back. So let's see how it happens. Why do we have lameness and how it happens? So this is the pathogenesis slide. This is our hypothesis, current hypothesis. Birds are growing really fast. Adding from around 25 grams per day to 100 grams per day. So this is a fast growth in those birds. This will lead to mechanical damage in the growth plate, followed by stress and immunosuppression, bacterial translocation. So in the gut, in those birds, in the intestine, we have what we call the intestinal tight junctions. They keep the intestine very tight. Once those birds are stressed, those tight junctions excuse me, will open, and the bacteria will leak from the gut to the bloodstream and eventually goes and settle in the long bones in the femoral and tibial heads. So bacterial translocation leading to bacteremia, followed by abscess, and finally BCO lameness. So what do we do? In my research program, and again, I currently have 6 doctoral students in my lab, 2 masters, and 9 undergraduate students doing research. And I just recently accepted Euro and accepted another student. Her name is Sauka. And I'm in the process of accepting 1 more student. And so why do I focus on Indonesian students? 4 years ago, a student from Indonesia, she was already a student on campus. She emailed me and said, I want to switch advisors. She didn't like her advisor as much. So she emailed me and said, would you accept me into your lab? Her name is Andy Asnayanti. She's, again, her family is from here. Actually, I had a nice dinner with them yesterday with her family here in the city. And so I accepted her into my lab, and I've seen nothing but the hardest working student I've seen in my life. Hardworking, polite, respectful. She defended her PhD under me back in April, so 6-7 months ago. She published 4 papers as a first author, 4 papers as a second author. So then I went to Andy and said, you're the hardest working student I've seen in my life. Can you help me accept more Indonesian students into my lab? So then she actually was able to contact Wuro, Sauka, and again, I'm in the process of accepting 1 more Indonesian student into my lab. And I want to say here how much I admire the culture, because I see it's a culture thing. People are very respectful, hardworking, and which is why my lab right now is the main lab that does this research in the entire United States. My main focus is lameness. So again, I want to see more research collaboration with the BRIN, with you guys, and also hoping to accept more students in the future. So what are we doing? So when birds go lame, you've seen I have the broom and I try to move the birds around. And the birds that are unable to stand, we pull them out, we sacrifice them, we kill them with CO2, and then we cut them open. When we cut those birds open, sometimes lameness is happening due to unknown reasons. We don't know they are lame, but we don't know why. But sometimes there are obvious causes, which is what I mentioned, kinky back or twisted legs. After we kill the birds, we put them in 2 categories. Either femoral head lesions, so femoral head separation, femoral head transitional, femoral head necrosis. Or we have tibial head necrosis, which is tibial head necrosis severe, tibial head necrosis cases. So we have a classification system that we use to diagnose those birds. I think one of the main issues, and I visited many countries looking for lameness. Every single time I ask, I get told that, we don't have as much lameness, or we have some cases. You cannot say that you have lameness or not until we cut those birds. And then we expose bones, femoral and tibial heads, and then we can see whether they have lameness or not. Why do we need to cut those birds? So chickens are very smart. There are many people, in fact, I'm from the Middle East, and they make all kinds of jokes on chickens being non-smart, stupid. But chickens are very smart. What they do is sometimes you go and, let's say we have 100 birds and we move them around, and you can easily say this bird is lame, this bird is not lame. Problem with those birds is that for the most part, they try to hide the pain. They are not showing the pain. So even though they have pain in their femoral and tibial heads, they pretend that they are okay. They walk normally without issues. Why? Because once they show weakness, other birds will be stumbling on them and they will kill them. It's like humans, correct? Don't show weakness. Once you start showing weakness, then others will be picking on you. It's like my kids in the school, they go and sometimes they come back and I was bullied in the school today. Well, once you show weakness, it's everywhere. Other kids will be picking on you. Same for chickens. They try to look strong and everything is fine, but they have too much pain inside. So why again? Because you can see right here, once this deer showed some weakness, the cheetah was able to pick that deer. So what's happening is those are tibial heads. And for tibial heads, you don't know if they have infection until you slice them. And so again, that's an invitation. If you guys have farms where I can visit and I can cut those bones. I can show you whether they have lameness or not. So this is the growth plate right here. And all this is filled with millions upon millions of bacterial cells. All this is very infected all the way, which is simply food safety issue, and this is not healthy. Now look right here. This is 1 day old chick, 1 day old. See right here? There is no femoral head. The whole femoral head is gone. Do you think this happened in 1 day? Or does it take time for this to happen? Do you think it happens in 1 day? This entire infection to eat the entire bone and there's no more femoral head? This takes time, at least 15 days, at least 2 weeks. So this tells us that the infection is actually happening vertically. So those chicks can get the infection from breeder lines or from the hatchery, and then the infection starts eating the bone. So on day 1, you see those chicks. In fact, in the US, we call them hatchery trash. Nobody labels them anything, but they're hatchery trash. Let's just throw them in trash. But they are infected. Other birds are infected as well, but their immunity can help them resist this disease. Those are my fingers right here. So this is a day-old chick. This is how a normal femoral head should look like. White, nice, shiny cap. Same thing right here. Look right here. Look right here. No femoral head. The whole entire femoral head, this entire femoral head is gone. No femoral head. And that's mainly because the bacteria will start eating the femoral head and causing the infection. So let's see how we do the diagnosis in my lab. Those parts are all lame. So this is a kinky back, this is kinky back, and this is just a lame bird. And again, kinky back is one form of lameness. This is a femoral head that's normal, shiny white cap. And if you go from here all the way down, this is from less severe to more severe. This is femoral head separation, separation of the cap. Femoral head separation. Then when the necrosis starts going deep, we call transitional, femoral head transitional, femoral head transitional. And then when the infection goes deeper, we call femoral head necrosis. Look right here. So I want you to compare this bone to this bone. This is the femoral head right here. No femoral head. Imagine if you're a human and you don't have a femoral head, how much pain? Humans, once you have some pain, oh, I just want to go to the doctor. Those birds, they try to hide it. They are not showing the lameness. Tibial heads, right here, you slice through the tibial head. This is normal. You press it with your finger. Solid, no issues. This is tibial head necrosis. This is tibial head necrosis severe when the infection hits the growth plate. And you can see right here, this is melted, liquid, entirely liquid because of the necrosis. This is what we call caseous, which is cheesy, basically. And this is what we call TD, tibial dyschondroplasia. So one other form is, again, as I said, kinky back happens whenever there is an infection in the thoracic vertebrae number 4 or flexible thoracic vertebrae right here, because this one is connected to the air sacs. Every single time those birds will breathe the air, all this air is actually filled with bacteria. I teach microbiology at the University of Arkansas, and my class is 470 students, 470. It's a big classroom. Now on day 1, I go and tell students, do you think when you breathe the air, does it have bacteria? And many students say, no bacteria, because they think there is a filtration system and it will filtrate bacteria. We're breathing. Every time you breathe in, it's filled with bacteria, correct? Our body, human body, has 6 to 10 times more bacteria than body cells. We do have 6 to 10 times more bacteria than body cells. So everyone, again, thinking it's, even when you go and buy chickens from the market, many people think that they don't have bacteria. No, those chickens are actually filled with bacteria. What do you think the best way to deal with when you buy chickens from the market? It may have salmonella or it may have something else. What's the best way to protect yourself and your family when buying chickens? What do? You eat them raw, cook them, correct? Cook them very well. So if you're grilling them on a charcoal, you need to have an internal temperature of 164 Fahrenheit. You're safe. Or if you're cooking, like you making a stew and all these delicious dishes, if you're cooking the chicken very well, then you should be safe, no issues. So right here again, birds will breathe the bacteria inside, gonna go into the air sacs, and eventually it will infect the thoracic vertebrae number 4. Now, as I said, if you go now and ask poultry producers here in Indonesia and say, do you have lameness? Oh, no, we have just a little bit of lameness, that much. but that's what they see, clinical lameness. Clinical lameness, what you don't see, the births hiding is that much. Most of the births will have lameness, but again, it's going to be in different degrees. Why is lameness important? Well, again, as I said, this is clinical, this is subclinical right here. Once you have lameness, there is a high incidence of mortality. Many birds will die. It's going to affect the feed conversion rate, body weight, uniformity, carcass quality, food safety issue, animal welfare or animal well-being, production, carcass quality. So lameness has a big impact on all those birds, and the financial losses caused by lameness, because once you have birds infected, their weight is not going to be the same. So if you have a bird that's 4 kilograms, birds that are lame maybe 3 kilograms or less. So you're losing money because of the weight. But how does it happen? How does this infection happen? Let's look right here. So when birds breathe the air, it's going to go to their lungs, and the bacteria will leak into the bloodstream. Or sometimes from the intestine, through the intestinal tight junctions, the bacteria will leak into the bloodstream. Or sometimes through the skin of the feet, also bacteria will leak. Once the bacteria leak, we'll have bacteremia, and then the bacteria will settle in those growth plate, long bones, fast growing birds, and eventually this will lead to infections, lameness. How to deal with lameness? So in the US, January 1st, 2017, the US said no more of mixing antibacterials, antibiotics with the feed. We used to mix them all the time. We mix them as growth promoters to keep the bacteria under control. But then the US said no more. Why? Because if you're feeding those chickens antibiotics, then this will increase antimicrobial, will make the bacteria more resistant in your body. No more. So then our focus either, so now we understand the etiology, we understand which bacteria. There is actually my lab. We don't only just deal with lameness, but we diagnose bacteria. So we do lots of bacterial sequencing, culturing bacteria. US said no more drugs, unless if you have a prescription from a vet and you have a severe case. So the focus now is on diets and vaccination. My lab, we developed a vaccine this past year, and the vaccine received a patent from the University of Arkansas. After you receive a patent, you have 1 year, and then they will submit that patent to the US Patent Office. So I'm hoping in the next 2-3 years, this patent will be approved by the US Patent Office, and it's a multibacterial vaccine. My focus is mostly on diets right now. We have so many companies that gives us their probiotics and prebiotics. I think I saw Zimpro, for example, in the slide from Dr. Harry. There are so many companies like Novonesis, Christian Hansen, Zimpro. All these companies give us diets. We mix them with the feed, and we try to see whether we have more or less lameness. So this is exactly what we have, and we're blessed that we have one of the best chicken centers in the United States. It's called Tyson Center of Excellence, where my lab is. And it's called the Tyson Center of Excellence because Tyson paid the university too much money so they can build the building and name it after Tyson. And so we have very nice farm where we have all these research trials are done. And so to study lameness, we had to create models. I need to induce lameness, make birds lame, and then I can give them a vaccine, probiotics or prebiotics to see which one of them is very helpful. So we created actually 3 models. My postdoc advisor, Bob Weidman, created this model. We call it the wire flooring model. Then we have the regular model, which we can see there are here carboys containers. We fill them with water, we add bacteria, and we make birds lame. And then we have what we call the hybrid model, which I will discuss shortly as well. So that's the carboys model. This is the wire flooring model. So you're raising birds on actually wire. This is how I induce lameness. You raise birds on wire, you put the feed on one side, you put the water on the other side. You're forcing the birds to walk back and forth between feed and water, back, forth, back and forth. And because the floor is very hard, this is going to create micro fractures in their femoral and tibial heads. It's going to stress them, open the intestinal tight junctions, and those birds will become lame. And this can create lameness of 70 to 80% of those birds will become lame. 70 to 80%. And so to use this model, you can see this pin right here and this one. 2 pins. Those are both wire flooring pins. The rest of the pens are actually just litter. So you can see those birds, they don't have wire. Only those two front pens, they have wire flooring. And each pen is 5 by 10 feet. We have 50 birds in each pen. Birds on those wire flooring model will become lame first. Once they become lame, they will breathe the bacteria that cause lameness into the air. We have tunnel fans here at the end of the house, and we have cooling pads. They're supposed to be right here and right here. So we're sucking the air from here all the way down, and all the bacteria is spread on every one of those birds, which will create lameness. Now, if we have 70 to 80% in those pens right here, usually other pens will get around 60, 50% of lameness. And you're giving them all different probiotics and prebiotics, and then you will see which one of them is actually helpful in reducing lameness. And so this is actually the model that we published this in the Poultry Science Journal, which is our #1 journal for poultry science in the US. And this design is actually by NDS Nayanti, the Indonesian postdoc. She's actually now my lab manager. After she graduated, I asked her if she's interested in becoming my lab manager. And she's actually the one who designed this. You can see right here, we have cooling pads here. We have tunnel fans at the end. Those birds on the wire flooring model will become lame. You can see the airflow is going this direction. And then those birds will become lame. You will see those birds becoming lame. And then again, you're giving them feed additives to see if feed additives can help reduce lameness. Most of the probiotics and prebiotics we use, anything that can help improve the gut health, immune health, we're always willing to try. So if you know a company, for example, here in Indonesia that have feed additives and they are interested in trying them to see if they can help with lameness, we're more than happy to try them as well. I work a lot with Europe and with Latin America. We don't have anyone in Southeast Asia or in Asia in general. We're more than happy to help if there is an interest. So you can see right here, all these bacteria right here in the gut. If we improve the health of the tight junctions, this will help reduce lameness. We do have a very long list of bacteria that cause lameness, from staph to enterococcus to E. coli to salmonella to strep. But we do have basically this bacteria, Staphylococcus agnatus, which I diagnosed when I was a doctoral student 10, 11 years ago. We published this in PLOS One. And this one, I was the first one to diagnose it in lame birds. Before that, it was only found on the hands of farmers that deal with cows that have mastitis. Again, we continue to work on publishing manuscripts related to trying probiotics and prebiotics and their impact on lameness. This is just part of the students. This is my group is actually expanding. That's NDS Nayanti, again, the Indonesian girl. We have, so Carla is from Saudi Arabia, Andy is from Indonesia, Anne is from Vietnam, Ravinda is from Sri Lanka, and Amanda, you can see with the cowboy hat, she's from the US. And then all these are my undergraduate students. Manal is actually a postdoc from Tunisia. And that's my group right here doing one of the experiments. And I want to open it for questions. And thank you so much for taking the time to listen to my presentation.
Thank you for very nice lecture, Dr. Adnan. So, Bapak dan Ibu, I think, would you like to sit here or? Oh, yes. Ya, Bapak dan Ibu, we already hear the lecture from Dr. Adnan. I think we can get the detailed information how important the BCO lameness for poultry production. So it's caused by we need the fast growth broiler to food safety and security, and then it will make the abnormality for leg. And I think before it just for degenerative disease, but it's also important for food safety because it will bacteremia and not good for consumption. I think we all of you have so many questions for Dr. Adnan. So if you have something that you need to clarify or you want to ask, you can directly raise your hand in Zoom, or you can type the question in the chat box. Or in this room, somebody want to ask to Dr. Adnan? Yeah, Dr. Hari.
Okay, thank you, Dr. Adnan Arubai, for very great presentations and new informations regarding to the Lameness. As you mentioned before, that it happens a lot in US. I think the production system here, probably in Broiler, between US and Indonesia probably different, particularly regarding to how long the farmers they manage the chickens. I think probably in US longer than in Indonesia. How does it influence the prevalence of the lameness in broiler chicken, and when normally the lameness found in broiler chicken? Does it like within 1 week, 2 weeks, or 3 weeks after cod infection? Thank you.
Very good question, Dr. Harry. So generally lameness starts, let's go back to your number one question, which is the difference between the Indonesian chicken industry and the US industry. So most chicken serving restaurants serve chickens that are somewhere between 42 to 45 days in the US. And there are certain, so the chicken production depends on what's your final end product. You want to sell only chicken breasts that are big. Sometimes they keep them up to 56 or even 60 days. But the ones that you can buy, for example, in the US, KFC or other restaurant food chains, it's somewhere between 42 to 45 days. Now, diets are different. So at 42 to 45, you can get somewhere between 4 to 5 pounds birds, which is a big size. That's mainly because of the differences in broiler lines, which lines you're using and which kind of feeds, and of course, the management of those flocks. The other question is, when does lameness start? So you can see from the chicks that I put on the screen that you can see lameness at day 1 all, because that's actually developed from the infection was transferred, or the infection can be transferred 2 ways, either vertical or horizontal. Vertical from breeder lines to chicks in the eggs, and you have 1 day old chick that is just hatched and has infection. Sometimes not breeder lines may be okay, but the hatchery. So I've been to hatcheries in the US that are very old, 50, 60 years. They are in bad shape. So when you put the eggs there, the eggs will get infected. Eggshells will be filled with bacterial pathogens. Those bacterial pathogens can penetrate the egg and go inside and infect the chick. But generally, lameness is a fast growth problem, and it's size related to problem. So there are many ways for birds to become lame, but the main way is once they get heavy. So maybe it's day 35, you don't see lameness, but that does not mean that the birds are not lame. They may have it. So if you look through the slide for the iceberg, most lameness that we see is actually only the tip of the iceberg. Most lameness is hidden, subclinical. And so one of the ways that I've been working with so many companies in different countries is we go there, and the first step we do is we do a survey. So let's say here in Jakarta, or just in, we select places in Indonesia, and you work with farmers, and you ask them to give you maybe 100 birds each, and you cut them open and then look for how much lameness you have. And then you design, you say, okay, well, this is and I looked online, there are no lameness studies here in Indonesia. So one of the ways we can do is, again, if we can find a way where we can buy those birds, I can come here, bring Wuro or Andy or anyone else with me, and then we can cut those. And of course, we'll pay for a flight tickets. It's just, whatever we have here, like buying the birds and all these things. Those are the things that we need to get some help with. But doing a survey study is the first thing every time. And then after the survey study, then you find ways of how can I improve my management practices, which probiotic I should use, which prebiotic. And of course, all those things must be bought locally. So there are companies that sell them and we need to try them. Plus we can try vaccines. But in general, as I said, many birds will have lameness. It's mostly subclinical. The birds that show clinical lameness are the ones that are very heavy. So after day 35, I don't know how long you raise the birds here in Indonesia. Yeah, so you may start seeing early clinical. We don't know about the subclinical until we cut them, but you can just simply do a study of clinical lameness and see how much lameness you have. And the diagnosis is easy. You just need to broom, and you just move the birds around, and the ones that are unable to stand or reluctant, those are the ones that are lame. So you can look for a clinical lameness, or you can look for subclinical lameness as well.
Thank you, Dr. Adnan. I also have a question related to vaccine. What is your strategy to develop the multibacterial vaccine related for BCO?
So good question as well. For the vaccine, over the years, as I said, my lab, we do the experiments in the farm, and then we culture bacteria from the bones. And over the years, we came up with a list of pathogens that mostly cause lameness, from staph, for example, different species of staph, E. coli, enterococcus. And so after years of work, I've been working on lameness since 2012, so over 12 years. Now we came up with a list, and we said those are the pathogens that mostly cause lameness. My colleague, Pami Jasud Hassan, he's originally from India. He works for the USDA, United States Department of Agriculture, and he uses a technology called electron beam, electron beam. And this technology is simple. So you have bacterial cells, and you shower them with electron beam. So you put the bacteria, shower them with electron beam, and the electron beam will destroy their DNA, shatter, damage their DNA, but they will still be alive. They cannot cause infection. They cannot multiply when you take them. So after he actually exposed them to E-beam, we take those bacteria, and again, we have a long list of bacterial cells that we mixed in the vaccine. You inject the vaccine at day 18 inside the eggs. So when chicks hatch, they already have some immunity. And then we found this vaccine to be very effective. But again, it depends. So we did this study based on the bacteria that we isolated from all of our trials in Arkansas. But let's say we want to do something here in Indonesia. First thing is first. First, you do a study on surveying how much lameness do we have. Do we have 10% of lameness here in Indonesia, or do we have 20% of, or 5%? And then we have to culture from those bones. So again, we have our own lab developed technique for highly aseptic collection of bone samples and bacterial diagnosis. So we will need help to do diagnosis of bacteria. We may have primers, and I don't know how the system here works, but we're happy to share our technique. Then you need to do a bacterial diagnosis, and then you're going to come up with a list. So we visited, let's say, 10 places in Indonesia, and then we did bacterial diagnosis, and then we have now 20 bacterial species. Those are the 20 bacterial species that we can use for the E-beam vaccine. And then you inject it into day 18, chicks, eggs, and then after that, once they hatch, they will have the immunity, and then this is how then it's very important that you start the immune response before they hatch to prepare them early on. Because once birds are grown and once they have the infection, it's very difficult to battle that infection. So the list should come from the bacteria that you have. And we don't know, here in Indonesia, again, I was so excited going online and trying to check. There are no lameness studies. As I said, my lab right now is the main lab that does lameness in the entire United States. So if we can do something, and I'm here again to propose research collaboration, if we can find a way to do surveying first, bacterial diagnosis, and we can, again, work on a vaccine or a probiotic prebiotic mix to see how much we can help the industry. I mean, at the end of the day, Indonesia is a large country, it's 270 million people, and you always need more protein. So that's why, again, the focus worldwide is on chickens. You don't want to raise cattle and wait for 3-4 years. Last year, I went to Kazakhstan, and I visited 3 places in Kazakhstan: Astana, Semey, and Kostanay. And I went to chicken farms, and I cut birds. And I don't know how many of you guys know, but you know what's the main meat in Kazakhstan? Anybody knows what's the main type of meat they like in Kazakhstan?
Goat.
Horses. Yeah. I didn't know that, but they like horses. So I was trying to convince them, look, and they already started, they already focusing more on, they want to focus more on chickens because horses is more expensive and it takes time to raise horses. Chickens, 35, 40 days, you can get those chickens. So at the end of the day, we did, again, a quick study with Kazakhstan, trying to survey Kazakhstan. So same thing can be done here. You can survey chickens, you can do bacterial diagnosis, and you can take it from there. And the more and the healthier those birds will get, you will get more meat production. So if you build good immunity early on and those birds are raised really good, you will get more meat production. Your chicken producers will make more money, the economy will be better, and people will eat healthier chickens.
Thank you, Doctor. So, yeah, next question. I got some question from the Zoom. First, from Mr. Gufroni. How to prevent the bacterial lameness? And the second, what should the organic producer do to apply the chicken management to deal with this bacterial disease? Thank you.
So chicken lameness, as I said, is because it's a very complicated problem. Chickens are not raised in a clean environment. You have chickens that are outside, so you're dealing with all different pathogens. And we call it in the US, there is no silver bullet. There is no magic solution to solve lameness. But there are things that we can do to reduce lameness. If you focus on improving gut health, so if you're using probiotics or prebiotics that improve gut health, more lactic acid, lactobacillus, or other microorganisms that improve gut health, anything that improves the immune health will be very helpful as well. So when you're preparing diets, mixing probiotics and prebiotics. For example, zinc is very helpful. Yeah, zinc, manganese, copper, really good. And then you have, those are prebiotics. Then you have probiotics. You have lactobacillus, for example, other microorganisms that improve the gut health. And then you mix probiotic with the prebiotic to get what we call symbiotics, which is a mix of probiotic and prebiotic. So mixing them can also improve both the gut health and the immune health. Every single time you're improving the quality of the diet, improving your management practices, you can reduce lameness. And then there's the factor of you need to know how much lameness you have. So you have a chicken house, and chicken house have water pipelines. Do you clean those water pipelines? Do you flush them? If you don't flush them, those water pipelines, you will have what we call biofilms built in. And biofilms, bacteria can stay inside and get protected. Nothing is going to happen. So you need to flush water pipelines, disinfect water pipelines. You need to check your birds and see, do you have lameness, how much lameness you have, and which bacteria you have. So in general, again, improving diet management practices can help reduce lameness. But again, before that, you need to know how big of a problem do you have. Do you have a big problem of lameness? Do you have a mild problem, or maybe you have very small problem in lameness? All those can be done with the diagnosis of the disease itself.
Thank you, Doctor. Related your answer about the mineral supplement like copper. So in my study, I found antibiotic resistant gene available in one plasmid with copper resistant gene. So do you think it's the same, sometimes bacteria need copper to live, right, doctor? But in this case, the resistance to heavy metal, it's same gene for this, how to say, I just afraid if we put the supplement contain the copper, then I will maintain the resistant plasmid. How do you think?
So there's a possible, it's a sword, you know, it's a two-edged. We call it a two-edged sword,
Yeah.
which means you may be helping the bacteria, but you're improving the host as well.
I see.
And our focus is every time we do these studies, our focus is on the bird.
Oh.
Can I improve the immunity of the bird? And then if the bird immunity is good, then they can beat the bacteria. So there is, again, copper is one of the minerals that we used. We use zinc, we use manganese. So even for humans, if you want to improve your immunity, you can actually take zinc tablets, which can help boost your immune system, makes you more resistant to infections. During COVID time, many people, started using lots of zinc because improves and strengthens the immune response. So back to your question, to your study, I think it's worth trying and comparing. But you tried it in bacteria, we need to focus more on chicken and just do a comparison. That's a good study to do as well. Scientists, we can make hypotheses, but we don't make clear cuts until you try it. So I think it's a good way of comparing bacterial response versus chicken response to the use of coppers. A good study.
Thank you for nice idea,
Of
doctor.
course.
Yeah, so in that, some side they think the microbiome is the main point to the health of the host.
Yeah.
But some side said the host immunity is important.
Yes.
But we need to keep the balance actually.
Yeah, so, as I said, for us, there are certain things that you can control. So you can control how much you add those feed additives in the diet. You can control how you can improve the gut health and immune function. But with the bacteria, it's like I always like to explain to my students in class, bacteria and humans or bacteria and animals, it's a battle, correct? We're trying to kill them and they're trying to kill us. So we always develop ways trying to kill them. They always develop ways to resist us because they are not going to sit there and wait for us to kill them, correct? It's a battle between us and there is no clear winning in this battle because bacteria always evolve and we always evolve as well. So penicillin, for example, penicillin was discovered 1928 by Fleming. And at that time, it was very powerful, very strong antimicrobial that can kill most bacterial cells. Nowadays, most bacteria is resistant to penicillin because of the development in antimicrobial resistance. So bacteria evolve, but we also evolve. We try to improve our ways so we can fight those bacterial pathogens.
Thank you, doctor.
Sure.
So how about in this room? Somebody want to ask Dr. Adnan? We still have question from Zoom, doctor.
Yes.
This from Brigita from Solo. How are chicken more susceptible and transmission? What the impact for the carcass?
What?
The impact for the carcass?
Well, the impact on the carcass is like what I discussed. Once those bones, so every time you have the tightened junctions open or the respiratory system leaking bacteria, then you have the bacteria in the bloodstream, bacteremia. When you have bacteremia, it means the bacteria is everywhere and every single tissue in the body will have this bacteria. So let's say you have the bacteria that's causing the infection is E. coli. E. coli is gram negative. Gram negative, they secrete endotoxins. If the bacteria is staphylococcus, staphylococcus has exotoxin. So literally, if you're allowing the bacteria to grow inside those birds, you're allowing the bacteria to produce toxins, endotoxin and exotoxin. And those are very damaging and they can have health impact. So for instance, people who get food poisoning from salmonella, what do they get food poisoning? What's the reason? The reason is the endotoxin. Endotoxin in salmonella is what causes food poisoning. So if you're allowing salmonella, E. coli, and staph to be in the carcass, it's going to affect the meat quality, how the meat will taste. Plus, it's going to have bad consequences. Let's say people will buy the chickens and they just grill them on charcoal, but they are not grilling them to perfection. Those bacteria can still survive and they can cause people to become sick, depending on which bacteria we're discussing. So lameness itself has an impact on the entire carcass quality, especially if it is extreme. So if you recall from the bones, if the bone femoral head is gone, and let's say you're trying to eat a femoral head and you find the femoral head itself is gone. Many people don't pay attention because you just eat and you think it's actually cooked because of cooking. Myself, before I allow my family to cook any bird, I'm holding my knife and just cutting. My wife don't like it, but I don't tell her sometimes. So it's very important that you keep those chickens healthy because that's going to impact the overall health of the bird. Otherwise, lameness has negative consequences on the carcass quality.
Thank you, Dokter. Ya, untuk Bapak atau Ibu Brigita, tadi informasinya dari Dr. Adnan bahwa lameness ini berpotensi menyebabkan bakterimia yang mana itu tidak lepas dari keamanan karkas tersebut itu sendiri. Karena banyak sekali faktor patogen yang akan dapat menjadi infeksi dari bakterimia ini. Dan tentu tadi yang dikatakan oleh Dr. Adnan, kalau di budaya di kita, karena dimasaknya matang, nah itu salah satu bentuk pencegahan kita agar tidak terinfeksi bakteri yang tadi. The next question from Dr. Riza. You want to directly ask him?
Thank you. My name is Riza Hartawan from Research Center for Veterinary Science. First, that you mentioned before that the infection could be from the vertical infection, and here in Indonesia we have for DOC we have several grades from the good one to the bad one. Usually the breeder keep the good one for their group and sell even the lowest grade DOC to the farmer. So I don't know the regulation in US. Did government punish the breeder to sell the low grade DOC to farmer? And if the farmer start their DOC with the lowest grade, do you think there is any mitigation like using the antibiotic or something because it's bacterial? For the second question, because I'm from virology, I know that some virus have effect not only lameness but paralytic. Like I study the Marek disease. I know in the US the most pathogen virus is in the US for Marek because the broiler is kept for a longer period in US, maybe for 56 days. Do they conduct vaccination for Marek or other disease like we know that Marek we vaccine in the breeder. So is in the US already start other vaccination beside the Marek in the breeder? I think this two questions is enough for this time. Thank you.
Now has more shares compared to crops of aviagen is crop. You need to sell the best birds to all farmers. There is a good system. single chick in the United States that received the Merck vaccine. December of 2023, we got hit. Law says, or like we have quarantine system. You have 2 million birds, you kill all of them. So that's a challenge.
Oh, yes, sure.
Thank you for the opportunity. So I want to ask about this vaccine development because it's very interesting. You made the vaccine and the application is you inject to the egg. So how long did you expect the immune response should develop before the infection occur? Because it's important that this chicken should have a immune response first, the antibody. There are also about this antibody. You expect the IgY, IgG, or IgA on that. That is important on this. Okay, thank you.
Very good question. So with the vaccine, so you cannot stop, so even like the drug. we start, the better. Because once you prepare that chip, there is a immune response. Fully decontaminated, there will be some bacteria. So once the bacteria lies on the shell, they can and try to protect the chip. Then if you're starting with the immune response here, once those chips hatch, that means will start to block the immune response. After they hatch, because they get water and eat. The immunity early on, and the vaccine is only one way. So I'm not then you have the great formula. It's a formula. It's not one thing. So you have 1, 2, 3, 4, 5. You mix them. Okay. So you have all these things that you're mixing from vaccine, prebiotics, probiotics. So why do I think prebiotics are important? So if you go to Europe, I gave multiple presentations in Europe. In fact, 4 months ago, I went to Belgium, and they brought like 20 veterinarians. I was training them on how to cut the chickens and look for the infection. And we discussed, again, what to mix with the diets. In Europe right now, they focus a lot on vitamin D, because you want to make that bone strong. So putting a right amount of calcium, right amount of vitamin D will be very helpful. Again, this is for the bone. So you want to improve the health of the bone, but you need to improve the health of the gut. You need to improve the immune response. So all these factors, plus again, giving the vaccine, I think, and that's why, again, I try to work on different feed additives, vaccines, because I think at the end of the day, hopefully in a few years, we'll find a way where this is the best way. You mix this much of probiotic, this prebiotics, this vaccine, and then you will get the magic formula. Yeah, it's a so because the disease itself is complicated, the response should be complicated as well, multifactorial. Sure.
Thank you, Dr. Adnan. We still have a question from Zoom from Ratri. Yes, we still have around 20 minutes left. How is the correlation between lameness and breed of birds? And then the second question, in your presentation, there is mention that the entry point of BCO causes by vertical transmission, intestinal and respiratory route. Is it any different of the length of incubation time? And in the other breed, like breeder chicken, there is many cases of lameness due to Staphylococcus. What is your suggestion to control it?
So as far as the breeds, we didn't find any differences between the different breeds that we use. So we use different breeds, and even we compared male versus female. Do we see more lameness in males or females? Of course, males will get heavier. So there is a possibility that you will see more lameness in male birds because they get heavier. But as far as the different breeds, we didn't see differences in the breeds that we have in the States, in the United States. Here in Indonesia, I don't know. Maybe there are some differences. So in science, you don't make a clear cut. There is no definitive answer because you always do research. So again, the answer for the breeds, and I don't know how many breeds you have here in Indonesia, but again, if I think it's worth a study, and again, you just came up with a different study that I would love to do as well. So not just the survey, but also comparing the different breeds. Each one of those can be a good study that can be published in a good journal as well. And as far as the second question was the Staphylococcus. Oh, we didn't do any studies on breeders, and the reason why is because the breeder lines are very expensive. So the poultry industry would not allow us to go and check their breeder lines for lameness, because each bird is extremely expensive. They don't want to lose any bird. Like if we go outside right now to a chicken farm and we buy 100 birds, it's not going to be as expensive as getting 100 breeder line birds. So we have not done any studies as of yet on breeder lines, but that would be something dependent on whether the industry would allow us to have access to their birds and check those birds for lameness. So for Staphylococcus, you need to do, of course, antibiotic susceptibility on those Staphylococcus to see what's the best antibiotic you need. You should not, with chickens, you should not use broad spectrum antibiotics. Because once you create those highly resistant antibiotic strains, it will be very difficult to get rid of them, specifically in the breeder lines. So first is check culture and find out which species of Staphylococcus. Is it Staph aureus? Is it Haecus? Is it Saprophyticus? Is it Agnatus? There is a long list of stuff. And then second, you do an antibiotic susceptibility. Third, one of the ways is, do you find the bacteria in bones or blood or tissues or all these places? But again, culturing the bacteria and doing antimicrobial susceptibility would be the best line. And now again, you should not use a broad spectrum, but also focus on, if you can do an in vitro study on staph, you bring the staph and see which, not just antibiotic, but what probiotic can help you control the staph as well. Oh, the flushing is always great. So with the flushing, you can use 10% bleach. So all water pipelines must be flushed with 10% bleach. You fill the water pipelines with 10% bleach and you leave it there for some time. You don't have birds. This is again when you don't have birds in the house. And then you remove all the bleach and then you run a quick water, like flush all the water pipelines with the fresh water. That's going to help you control the biofilms, because if you have a Staphylococcus, it will be, because birds will drink and the staph can simply hide in the biofilm of those water pipelines. But if you flush them with 10% depletion, if you go to veterinary supply companies, they also have, again, disinfectants that you can use on water pipelines. Flushing them and then running clean water through them, that will be very helpful as well. So management practices.
Yeah, thank you for good knowledge for us, Dokter. Mungkin Pak Agus ada, misalnya Dr. Agus, you shall have question.
I have a question on your iceberg phenomena. What do you think the burden of this disease, Dr. Dalton? Because when you're talking about, and that's one question. Second is that talking about vaccine, as Dr. Eddie mentioned before, it means the vaccine is really specific location for Indonesia is different with other, because even Indonesia probably different like ecosystem, different the agent, and then different vaccine also. What do you think?
So the iceberg phenomena, its impact is, again, the problem is you only see small percentage of lameness. Most lameness is hidden, which means if you have, let's say, 2-3% of lameness, you should not be happy, because you may have 20% subclinical. So you need to find out the focus of this, or many scientists focus on the clinical one. I personally want people to focus more also on the subclinical. Because if you have subclinical, that's going to impact food safety, animal welfare, feed conversion rate, the animal well-being as well.
Is there any calculation on that? I mean,
there is. So
currently WA is also preparing for burden because of disease and management because of the infectious and management thing. So when we are talking about the lameness, how about the non-infectious thing of the burden of the disease, of the phenomena?
So again, my focus is on the infectious causes of lameness. But again, at the end of the day, once you have lameness, and whether it is a clinical or subclinical, you will be impacting the quality of the bird, quality of the meat, the pain you're causing to the bird itself. And nowadays, nowadays there is more focus on making sure that those birds don't have pain. Because if you're raising the bird and then the bird is having pain, that's not going to be helpful. Birds, they have feelings. They feel the pain. And even for us as Muslims, I remember, again, there's a story that's known for all Muslims that the Prophet ﷺ said one time he saw a woman, and this woman found a stray cat. And she got the stray cat, brought the cat home and saved the cat. And he said she's going to enter heaven. She'll go to Jannah. Why? Because she saved the cat. So big part of our religion as Muslims is we should care for animals. And if they have pain and we can save them the pain and suffering, that's something that has lots of hasanat as for us as Muslims. So that's, again, when it comes to the, again, the iceberg phenomenon. What was your second question? And the vaccine.
If the vaccine was going to be registered here,
yep,
it means we have to challenge in Indonesian system, in Indonesian bacteria situation. Yeah. What do you think?
So for us, again, the vaccine we did, it took us like 3-4 years to develop the vaccine. And then we pushed the vaccine, we submitted the patent through the university because everything I do is owned by the university. I don't own it myself because I work for the University of Arkansas. And so we went through the patent process and we received they call it provisional patent by the university. And then this April, it will be filed through the US Patent Office. Once it's patented through the US Patent Office, then the university can sell it. Can sell the right, for example, to Indonesia. But to me, again, I think it's worth it. And it's more important that you find out, because the vaccine is going to have a selected list of bacteria that is based on the US. So to me, it's very important, we can sell in the university. I don't make the money, it's going to be the university making the money. But at the end of the day, it's a good start in the right direction. To me, this vaccine will continue to evolve. So if I come, say, for example, we come to Indonesia and we find, I guarantee you, there will be certain bacterial species that you find in Indonesia, Iraq, US, Saudi Arabia, Vietnam, China, everywhere. But there will be some differences as well. So you go to farm A and you'll find maybe more E. coli. You don't find staph, but maybe you find enterococcus. You go to another farm, you find more staph and more enterococcus. So it depends. But here in Indonesia, if you want to develop a vaccine, so once this is again produced and once it's patented, and you want to do the same thing here in Indonesia, of course, that's something that can be discussed down the road. But before that, it's very important that you do a full study in different regions of Indonesia to find mapping. You need to map Indonesia first. So we did mapping in ourselves, and again, there are plenty of references on lameness in region A in the US versus B versus. Our vaccine is again more to our system. You want to develop something here, you need to find out exactly what you have.
Bapak Ibu yang di ruangan, apakah ada yang ingin bertanya? Untuk Bapak yang raise hand, Purnomo Edi, ini bisa ditampilkan. Bisa ditampilkan. Silakan Bapak Purnomo Edi. Excuse me, Dr. Adnan. Maybe this is the last question from our audience. Directly want to ask to you from the Zoom. Ask to unmute.
Marketing mau izin nanti jam.
Silakan Bapak Purnomo. Mr. Purnomo. Okay. Yeah, I think it's there is trouble. Okay, Doctor. For the last question, maybe also from the Zoom. Is possible to how to say about the co-infection between parasite to as a cause of lameness?
I've never seen parasites cause does not mean that parasites are not going to the overall immunity of the bird and health, which can in return cause lameness, but not directly because again, it's bacterial chondro necrosis or osteomyelitis. But do viruses cause lameness? Yes, they do. Reovirus, for example, causes lameness. Parasites, it's kind of difficult because again, you get to the bloodstream, you need to get access to the growth plate of the bone, which is difficult for parasites because of their size. So it's, I would say 90% bacterial, and then there are other factors as well that contribute, but parasites can contribute indirectly to lameness.
Okay, thank you, Doctor. This is the last question. So, Bapak dan Ibu, ladies and gentlemen, we are already here the comprehensive discussion with Dr. Adnan. We hope we can get continue lecture from him for the future, and I think another researcher in our institute also have many questions, Doctor. Maybe we will contact you by email directly if we have some idea that want to share to you to collaborate, and yeah, we hope the support.
Anyone, if you want to get in touch with me, I can give you my QR code. I know you guys here in Indonesia are big on QR codes, or you can get my card, and I'm more than happy to talk with you. But again, thank you all for taking the time to listen to me. It's been a pleasure being here today.
Okay, I will send back the seminar to MC Ibu Yeti.
Baik, terima kasih Bapak Edi Mawinata yang telah memandu sesi pemaparan dan diskusi. And thank you very much for Dr. Adnan for the interesting presentation. Baik Bapak Ibu, cara berikutnya yaitu penyerahan plakat kepada narasumber pada hari ini oleh Kepala Pusat Riset Veteriner pada Dr. Hewan Harimurti Nur Aji PhD. Kami silakan. Kita berikan aplaus. Baik, Bapak Ibu, acara selanjutnya yaitu penutupan oleh Kepala Pusat Riset Veteriner pada Bapak Arif Indonesia. Baik, terima kasih Bapak telah menutup acara pada hari ini. Baik Bapak Ibu yang berbahagia, Alhamdulillah kita telah sampai di penghujung acara. Terima kasih kepada seluruh peserta webinar yang telah menyimak dari awal hingga akhir acara. Kami mengingatkan kembali kepada Bapak Ibu untuk isi presentasi yang sudah di-share oleh panitia di kolom chat. Saya Yati Listriani, mewakili panitia yang bertugas. Mohon maaf atas kekurangan dan kesalahan selama acara berlangsung. Izinkan saya menutup acara dengan sebuah Loh. Siang hari makan kuaci. Makan bareng sama ibu terkasih. Cukup sekian guest lecture hari ini. Cukup sekian dan terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Terima kasih Bapak Ibu.